So today we are going to dive into one of the coolest regulatory systems in all of biology. Seriously, it's this tiny little genetic circuit that, it turns out, is the engine behind a huge chunk of modern biotech. And the whole thing is driven by a molecular race against time.
All right, so think about this for a second. Pretty much all life on Earth, right? From the simplest bacteria all the way up to us, we all need these special proteins to kickstart DNA replication. But then you've got this one famous plasmid from E. coli called coli-1 that just doesn't. It completely controls its own replication using nothing but two tiny little bits of RNA. So the big question is, how on earth does it do that?
Okay, to really get this, we first need to meet the key players. You can think of them as the two main characters in our story. The entire system, literally everything, hinges on the interaction between two specific RNA molecules. We call them RNAi and RNA2. And what's really clever is that they're both made from the very same piece of DNA on the plasmid, they're just transcribed in opposite directions.
So let's break down the differences. On one hand, you have RNAi2. This one is the big one. It's about 550 nucleotides long, and its whole purpose is to become the primer that kicks off DNA replication. But on the other side, you have RNAi. It's much smaller, only about 108 nucleotides, and it has one single solitary job: to stop RNAi2 in its tracks. And the way it does this is by being perfectly antisense to the start of RNAi2, meaning its sequence is a perfect match, a perfect complement. So that's really the heart of it, right? You can just boil it all down to a competition. It's a straight-up fight. You've got RNA2 trying to get replication going, and you've got RNAi trying to shut it down. Everything else we're going to talk about is just the story of that conflict.
Right, and what that means is that for every single RNA polymerase II molecule that gets made, there are only two possible fates. It's like a fork in the road. It either succeeds and you get a new copy of the plasmid, or it fails and replication gets blocked completely.
That's exactly right. And this is a really important point. This isn't like a simple light switch, on or off. It's what we call a kinetic competition. The fate of that plasmid literally comes down to a race. Who gets there first? Does RNAi manage to bind to the DNA, or does RNAi catch it and intercept it before it has a chance?
All right, so let's walk down that first path. Let's look at what has to go right for the plasmid to actually get copied. Okay, it's basically a four-step process. First up, RNAi gets transcribed. Now, while it's being made, its front end starts to fold up into these three specific little stem loop shapes. Now, if nothing interferes, the other end of that RNAi molecule, the tail end, will actually invade the DNA double helix. It pries it open and forms a stable hybrid. That's when a host enzyme called RNase H comes in and makes a cut in the RNA. This creates the perfect starting line, the primer. And then finally, DNA polymerase I can latch on and start making a new copy of the plasmid. And this structure, this intermediate, has a special name. It's called an R loop. And you have to understand it is absolutely crucial. The whole point of those folded stem loops at the front is to position the tail end perfectly so it can pry open the DNA and form that stable RNA-DNA hybrid. If you don't get an R loop, the entire process just stops. Dead. And that's exactly where RNase H comes into play. You can think of this enzyme as a very specialized pair of molecular scissors. It has one job. It only cuts RNA if that RNA is part of an RNA-DNA hybrid. So it sees the R loop, it recognizes it, makes one single, precise cut, and that creates the perfect starting point, what we call a free 3' hydroxyl end, which is exactly what DNA polymerase needs to get to work. Okay, hold on, let me just make sure I've got this. So if RNA2 wins the race, it folds up the right way, it makes that special R loop thing with the DNA, it gets snipped by RNase H, and boom! DNA polymerase gets the green light and you get a new plasmid. Okay, simple enough. But what happens if it loses the race?
Well, that brings us right to our second path. What happens when our inhibitor RNAi actually gets involved? And trust me, this is where things get really, really clever.
The inhibitory pathway is every bit as precise as the replication one, and it happens fast, while RNA2 is still being transcribed. A nearby RNAi molecule bumps into it, and their loops, which are perfectly complementary, bind together. This first little touch point is called kissing. Once that happens, the contact gets stabilized, and this forces RNA2 into a totally different shape, a shape that makes it impossible for it to form that R loop. And if you have no R loop, well, you get no cut from RNAh, no primer, and absolutely no replication. And this is where we get that fantastic term, the kissing complex. The best way to picture it is to imagine two puzzle pieces that only connect at their very tips at first. The loops on the RNAi and RNA2 hairpins are a perfect match, so they just stick together for a moment. That initial kiss is the very first step to shutting the whole system down. Now, that initial kiss on its own is pretty weak. It doesn't last long. So to make it permanent, another player has to enter the scene, the ROM protein. It's this tiny little protein, a homodimer, and it acts just like a molecular clamp. Now, here's the cool part. It only recognizes and binds to the kissing complex itself. It won't bind to RNAi or RNA2 on their own. And when it clamps on, it increases the stability of that inhibitory complex by about 10 times, basically locking RNAi into the off position.
Okay, so what is the point of all this complexity? Well, it all comes together to create this unbelievably elegant system for controlling the number of plasmid copies inside the cell. The best analogy really is to think of it like a thermostat for DNA. The logic here is a classic negative feedback loop. See, the gene that makes RNAi is located on the plasmid itself. So let's say the number of plasmids starts to get too high. Well, that means the concentration of RNAi in the cell also goes up. More RNAi means more inhibition, which naturally slows down replication. And the reverse is also true. If the copy number gets too low, the RNAi concentration drops, inhibition is less likely to happen, and replication speeds back up. The system just automatically corrects itself. It's brilliant. And this chart really drives home just how important that little ROM protein is. You see, in a normal cell that has the ROM protein, the plasmid number stays right around 20 copies. But if you get rid of it, well, that kissing complex is much less stable. That means the system needs way more RNAi floating around to work effectively. So the copy number has to skyrocket to around 40 before that feedback loop really starts to bite. All ROM does is make the entire thermostat much more sensitive and much more efficient. So why should you care about all this? Well, here's why. This exact Col1 replication system is the engine inside literally thousands of different plasmids that are used in biotech labs every single day all over the world. If you've ever touched a PUC vector in a lab, guess what? You've used this system. And beyond that, it's historically huge. This was the very first example of antisense RNA regulation ever discovered. It basically opened up an entire new field of biology. All right, let's boil all of this down to the key exam points. If you walk away with nothing else, make sure you've got these three things locked in. Okay, takeaway number 1. RNAPII is the primer, but it can only do its job after it successfully invades the DNA to form that R loop. Only then can the enzyme RNase H come in and process it into a working starting block for replication.
Takeaway number 2. The inhibitor, RNAi, stops replication cold by catching RNA2. It uses that kissing interaction between their loops to trap RNA2 in a shape that makes it physically impossible to form that all-important R loop.
And finally, takeaway number 3, and this is maybe the most important idea of all. The whole thing is a race. It is a kinetic battle between two opposing events. The winner isn't decided by a simple on-off switch, but by things like concentration and pure probability. And that leaves us with one last thought to chew on. The Col1 system proves that life can make really complex decisions without involving big, clunky proteins, just by using the simple physics of how RNA folds. And it just makes you wonder, doesn't it? What other incredible biological switches might be out there, hiding in plain sight, controlled not by proteins, but by the simple, beautiful, and elegant folding of RNA?